Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today the video is related to RNA sequencing data analysis, and the title of my today's video is Understanding Reasons Behind Compromised Quality of Sequence Read. As you all know that I have started a video series related to RNA-Seq data analysis, and this is the part 5th of that video series. In the previous video, I have discussed the structure of the sequencing files, and I briefly talked about the quality of the files. But in this video, I will discuss the reasons behind the compromised quality of sequencing files. Okay, so this is very much important video, and after watching this video, you will be able to know that why the quality of the sequencing files gets compromised. Okay, and when you get the knowledge of it, so then you can deal with it and you can pre-process your data in a very effective way. Okay, so briefly discussing that I have started this video tutorial series by starting from the experimental design or experimental workflow. Then I told you about that what is sequencing, how it is performed, and now we are understanding the sequencing files. Okay, so this is the in understanding the sequencing file, you should know about the structure of it, the quality of it, how, why, or what the problems behind the compromised quality. So after managing to get well known about these steps, then you can easily proceed with the sequencing data analysis. Okay, so just start from the first part of this video series, and everything is in a flow. Okay, so you can learn in a very effective way. Okay, so now topics of discussion is that some of the problems in NGS data include adapter contamination, ambiguous base calls, poor quality bases, very short reads. Okay, so I will discuss first about the ambiguous and the poor quality bases, then very short reads and adapter contamination, and after that the host reads. Okay, so these are the main problems that are associated with the quality of the sequencing files. Okay, now just a bit recap of it, that before understanding ambiguous base calls and poor quality reads, you should know that what is sequencing. So I have discussed a lot about sequencing in the previous videos, but first we prepare the library. After that, then we just perform the library cluster generation, and then we perform sequencing. Okay, and as I have told you that after we are performing the sequencing, so we can say that there is a laser, and after that laser is scanning that fluorescence, and it is feeding that information into the sequencing files. Okay, so if we zoom into it, so when the laser is scanning that scanning that fluorescence light, okay, so just look here, there are millions of fluorescence that are simultaneously being produced by the sequencer. Okay, and the laser is scanning it the laser is scanning it, and then the reads are just written in that sequencing files. Okay, so for example, if we are looking into this specific point, okay, so when the green fluorescence is the thymine, after that it is guanine, cytosine, thymine, adenine, cytosine. Okay, so a single FASTQ file is being generated. Okay, so inside the FASTQ file, the sequence number 1 is being generated. Okay, so it's just like that. Okay, so the scanner is scanning it. Okay, so for example, this is the laser, or we can say this is the scanner, and after that it is just detecting the fluorescence. Okay, so what happens that millions of fluorescence are being produced simultaneously, so there is a chance that the scanner gets confused. Okay, so when it gets confused, so it just gives that that base a poor quality or a compromised quality. Okay, so for example, if it is the scanner is confident about that, yes, this is thymine, so the more the confidence, the better will be the score. Okay, so now there is also a chance that the sequencer cannot get the sufficient information about that, what is basically either it is thymine, guanine, cytosine, or that thymine. So just it give it n. Okay, and this n is basically ambiguous. Okay, ambiguous base call. Okay, this is the ambiguous base call. So if for example it is performing sequencing, and it is reading that sequences into the file, for example there is thymine, it read that thymine, then guanine, then cytosine, and after that there is thymine, but it is not confident about it. Okay, because of these specific mechanisms, so because of the confusion, so what happens that it gives this thymine ambiguous. Okay, so the scanner is saying that I'm not confident about it. Okay, completely. Okay, so if if the scanner is a little bit confident, so it give it it just write that sequence the base down, but if it is not totally not confident about it, so just it adds n to that sequence. Okay, and after that we can proceed with the adenine and cytosine. Okay, so each and every base have a quality. Okay, I have discussed this quality in the previous video. Okay, so just link that video with this tutorial. Okay, so first watch that video, then you can easily understand with this. Okay, so each and every base have a quality. So basically these are the ambiguous base calls, and it is not good. Okay, because when we are aligning our actual reads with that reference genome, so ambiguous base calls are not there in that our reference genome, so then there will be such type of errors that we are not understanding that what basically this n means. Okay, so this is basically about the ambiguous base call. A small explanation that why it is giving it. So I have told you that millions of colors are being produced and scanner is scanning it, but for example, if at a time in a cluster there is also green, there is yellow, there is red, so when we just add mix green and yellow, so different colors produce. Okay, So the scanner does not know about that what is basically the other color. Okay, it has only information about the green, yellow, blue and red color. Okay, so that's why when the color mixes up, so it get confused. Okay, so this is the basic thing. Okay, so we get ambiguous base calls. There is also qualities of poor, there is also reads of poor quality. Okay, it just detect that yes, this is cytosine, for example, but it gives it cytosine, but there is a chance that for example when it get confused, but it is not totally confused just like ambiguous base call, but it is not, the scanner is not confident about it, so it gives it a score. For example, out of 100 it gives it 10 out of 100. Okay, so 10 is a very much less score, so because of the confidence score, so the scanner said that yes, this is cytosine, but I'm not very much confident about it. Okay, so this is poor quality reads, okay, or poor base call. Okay, so there are ambiguous base calls and a poor base call. So in the upcoming videos I will then visualize these ambiguous base calls and the poor base, quality reads, and after that then we will just filter out these. Okay, so first of all you should know about that why does these appears and what are basically n this poor quality. Okay, so then we remove these reads because when we are aligning with the reference genome, so then there will be a problem. Okay, so just know about that what is basically ambiguous base calls and poor quality reads. Okay, now coming to the adapter contamination and short reads. Okay, this is very much important concept and the data pre-processing, because if you are not confident about adapter contamination and short reads, so there will be a problem in the pre-processing and you will get confused that how should I pre-process my sequencing files. Okay, so first of all, important concept, there are many sequencers, the iSeq 100, the mini-seq, the MySeq. I think you have heard about it, and if you are new to it, so I'm telling you that there are different type of sequencers that are sequencing our DNA sequences. Okay, so it depends. It start from the iSeq 100. It has the lowest power. Okay, it can give the max output of 1.2 GB. Okay, or gigabase pairs. Okay, then after that, mini-seq, MySeq, next-seq. I want to just look into this max read length. Okay, this max read length. Okay, for example, in the case of iSeq, when it is sequencing the reads, okay, so it can sequence about up to 150 base pairs. Okay, when it is sequencing our reads, okay, so up to 150 it can just sequence our sequence read. Okay, after that it cannot sequence it. Okay, so 150 is the length of the output. Okay, the max read length, it can start from 50, it can sequence 50, 60, 70, but the maximum is the 150 bases. Okay, when it adds 150 bases, then it's terminated. Okay, so this 2 means that 2 reads. Okay, or it can go up to 300 also. Okay, so this is 150. Now MySeq have high power. Okay, so it can just go through 300. So when it is generating the sequence, okay, so it can proceed up to maximally 150 base pairs. Okay, here in the case of 300 it can just proceed to 300 base pairs. Okay, so just keep this concept in mind. This will help you in the next slide. Okay, so the max read length of each and every sequencer is different, and 150 base pair and 300 base pairs are common. Okay, there are also 250. Okay, so this is the maximum read length, the power of the sequencers. Okay, so you should know about it. After that, there is an important concept that we previously discussed. This all concept I'm just providing you just to understand the adapter contamination. Okay, but first of all you should know about these concepts. Okay, the second concept is when we were just forming our library, the library preparation in the previous video. Okay, so this was the library preparation. Okay, before sequencing we have to just form our library. Okay, so I have talked about the library preparation. You can just watch the previous videos. Okay, so in the private library preparation, when we get our messenger RNA, so then we just convert it into complementary DNA, and then we perform fragmentation. Okay, so fragmentation is basically not linear or not uniform. There may be fragments of 50 base pairs, there may be fragments of 100 base pairs, there may be of 200 base pairs. Okay, so these are not uniform. There are different type of fragmentation lengths. Okay, so in the fragmentation there are different types of different number of base pairs. Okay, different number of fragments. Okay, so keep this thing in mind. Okay, so now as I have also discussed about the adapters, that what are adapters in the previous video. So basically this up to this green and red, or we can say this part is the adapter. Okay, and after that, this is the adapter. Okay, and this black color is our sequence of interest, and we want to sequence it. Okay, so yes, this is the flow cell. Okay, this is the base of the flow cell. Okay, now our sequence is attached and we want to sequence this black color. Okay, so what happens, so this is the primer binding region. Okay, so our primer will bind to it, and after the sequencing will be performed. Okay, so for example, if this is our this black color, for example, this is our 150 base pair of the fragment. Okay, so the adapters are being attached to it, and after that, just like here, the adapters are attached to ligation, and after the sequencing is performed. So for example, if this black color region is our sequence of interest, and this is 150 base pair, so after that the sequencer will start sequencing it, and when it reaches this part, okay, so it will terminate. Okay, because the maximum output is the 150 base pair for a sequencer. Okay, so when it reaches here, so it will give the sequence of our black region, and this will be 150 base pair. Okay, So yes, we can understand this. Okay. Now, what if when we perform fragmentation and this black color region is, for example, of 40 base pair? Okay. So when the sequence, and for example, now it is 40 base pair. Okay, 40 base pair. This is just because of the fragmentation. As I have told you that the fragmentation is not linear, not uniform. Okay, there may be different type of fragments. Okay, different lengths of fragment. So for example, now this is 40 base pair. Now when the sequencer starts sequencing, it binds to the primer binding region, and after that it starts sequencing. So when it reaches 40, so it will not terminate. It will start. It will continue its sequencing. So it will also sequence the adapter. Okay, it will also sequence the adapter. So for example, now this is our read. This is 40 base pairs. Okay, this is our actual read, but it also sequence the adapter. For example, adapter is, for example, also 50 reads. Okay, 50 base pair. So now this one is the adapter, and we do not need it. We did not need this adapter in our sequence because it is man-made. It is not the pure sequence, the actual messenger RNA sequence. So these are the adapters. So this is basically known as adapter contamination. So when we are having short reads, so this can happen because, so when we are pre-processing it our data, so we should remove adapters. Okay, because there is maximum chance of having adapters attached to our sequence of interest. Okay, because of this limitation in the fragmentation, because fragmentation is not uniform. Okay, so this was the concept about the adapter contamination. So we have to remove in the pre-processing step when we are performing the data analysis. Okay, so I hope you understand this concept of adapter contamination. Now there is also a problem of very short reads when we perform fragmentation. Okay, and after that when the adapters are being just attached to it, and when we remove the adapter, so just 40 base pair of sequence is remained. And now this is a problem because very short reads are not effective in alignment. Okay, so very short read is also a problem. Okay, so when we are pre-processing it, so we just tell the software or the tool that we need sequencing that start from, that have a length of 150 base pair or that have a length of 100 base pair. Okay, because these are the effective sequences. Okay, so very short reads just generate because of the fragmentation. It also generate because of when the adapters are being removed. Another important thing, that there is also a chance that the sequencer just terminate before the time. Okay, when for example it is having 150 base pair of maximum read, and it just terminate because of the technical reason or because of the insufficient reagents, it terminates, or different other reason, it terminates in from 50, and it just sequence this part. Okay, and this part is remembered. So this, for example, this part is 50 base pairs. So now this is also a problem. This is short read. Okay, so it can just manipulate our results. Okay, in the form of alignment and this type of thing. So basically this was the adapter contamination and the short reads problem. So how short is very short? According to the literature, 50 base pairs, less than 50 base pair is short. Okay, so less than 50 base pair is short. Our professors recommend 70 base pairs. Okay, that start from the 70 base pair or 80 base pair, but I have seen 50 base pair. But recommendation is that just take that reads that have 50, 70 base pair of length. Okay, so this was all about the short reads and the adapter contamination. Now a little bit we should talk about the host read. For example, we are sequencing a metagenome. Okay, metagenomes are basically bacterial reads. Okay, and when we are just performing metagenomics, and after that in the data we are also having the human reads. Okay, so human reads are basically host reads, and we do not need it. Okay, so we have to remove these reads. Okay, so this is our type of host reads. But metagenome, when we are just sampling it from the gut microbiota or so there is a chance that our actual human cells are also being captured, and after that the DNA is extracted. Okay, so there will be a chance that human messenger RNA is also present in the the RNA of bacteria. Okay, so we have to remove this human reads. Okay, so this is also a type of problem in NGS data. Okay, so this was all about today's video. I hope you got the point that what is the problem associated with NGS data analysis. The reason of just explaining this video before the data analysis was that when I will be performing the pre-processing step, so you should know about these concepts. Okay, so then there you will be confident that which type of flag or which type of parameter should I use to just pre-process my data, pre-process my reads. Okay, so stay tuned. Okay, you will get interesting videos. So topics for the next tutorial is the exploring biological databases. We will explore NCBI SRA database, sequence read archive. I will also explain about the European nucleotide archive or ENA database, and I will also explain the array express, because now we are confident about each and everything. We have just understand our FASTQ files, we have understand the structure of it, the quality of it, the sequence flow, the workflow of RNA-Seq. So now we are just diving into that. How should we just retrieve our FASTQ files, our data of interest? Okay, so basically secondary data is present in these biological databases, because we are now going to perform data analysis. Most of the people, Western communities, they just generate their primary data. Okay, and they just perform their thesis or just perform their research. But most of the researchers also perform data analysis on the present data that is present on these biological databases. Okay, so we are going to use these databases, retrieve our data, and start reprocessing of it. Okay, so now we are diving into the data analysis step. I think this theory is enough for you people. In the upcoming videos there will be theoretical stuff, but we will mainly focus on the practical stuff more. Okay, there will be a bit theory about the different steps, but the most important will be the practical steps. Okay, so stay tuned for more. Inshallah, you will get more interesting videos related to bioinformatics, related to RNA-seq data analysis. Okay, so see you in the next video. If you have any problem associated with the compromised quality of sequence reads, so this you can comment me in the comment box. I will answer you there, or you can email me. My email is present in the description box, so you can email me and you can tell me that what is the problem you are facing. Okay, so just be online and I will discuss each and every point. Okay, if you also need online assistance, so do let me know. I also provide online services to my viewers. Okay, so thank you very much for the today's video. If you understand it well, so just like it, subscribe to my channel, and also share it with your friends so that they could get benefit from it. Okay, so thank you very much. God bless you.